Awesome. So, Brook and John, thank you so much for joining us today. I'm very excited about this crossover between the Boston Computation Club, i.e., my group of nerdy friends from Boston who sip coffee and talk about category theory all day, and the broader runner world of Colorado, which is focused primarily on bioinformatics. So we'll be hosting John and Brooke, who are both PhD students in uh, the UC Anschutz or Denver system, and learning about causal inference. And John has the rare privilege of being not just a great academic, but also my housemate, which is, I think, a first in the history of the Boston Computation Club, and probably the most important fact to know about him. So with that, I'll let you two take it away. Thank you so much for joining us.
Cool. Yeah, thanks, Max. Like you said, I'm John. Brooke is also here.
Hi,
Uh,
everyone.
and yeah, and we're both PhD candidates. In our last year, we're both in the Lowsbone lab, which is at CU Anschutz. And talk is kind of what would happen if you took our two independent dissertations and smushed them together and something were to come out of it. Um, yeah. Do you have anything else to add on that, Brooke?
I don't think so. Let's get at it.
Cool. Yeah, and so yeah, I work a little bit more on the statistical side of studying the gut microbiome, and Brooke also works more on the knowledge graph side of how the gut microbiome interacts with human health. And so that's what we'll be talking about is kind of these biomedical applications towards the gut microbiome. And just to give an outline, I'll talk a little bit about what causal inference is and how we approach it. Brooke will talk some about knowledge graphs and how we use them and construct them. And then because we're both focused on the gut microbiome in the Lowsbone lab, we'll talk about methods for understanding the gut microbiome and what questions we might be interested in asking. And then we'll talk about using knowledge graphs to increase statistical power and complement causal inference type work. And kind of after each of these sections, we can take like 1 to 2 questions, and we'll have some time for questions at the end. But I'll jump in, and I just want to talk about how understanding causal mechanisms can be really useful. Because there are a lot of questions that we're interested in asking, where we want to know how causal mechanisms work, and we want to be able to think about interventions. So, for example, Max could like to smoke cigarettes, but he doesn't want lung cancer. And he might be interested in questions like, will I get lung cancer if I smoke cigarette? And in this case, it's very interested in some interventional question. And uh, typical methods would maybe go about looking at association-based testing on other people. So if we take 1,000 people, what is the likelihood that someone has lung cancer given that they smoke? Or we might do some intervention where we make someone smoke and see if they get lung cancer. But even in that situation, there's a problem because we don't know if they would have already gotten lung cancer in the other situation. And with the association-based testing, we also don't know that's just correlational, it is not causal. And traditional statistics relies on association testing, like what is probability of cancer given that someone smokes. But we're really interested more in approximating counterfactual scenarios. And that's what the field of causal inference is geared towards. So what is the probability of cancer given that we intervene on smoking? And this is a difficult question to answer because there are a lot of variables like your personality and the environment that you live in that both affect whether or not you will smoke and whether or not you will get cancer. Um, and a lot of causal inference revolves around controlling for the appropriate variables to let us approximate these counterfactual scenarios. So we want to take this situation where we have smoking, which impacts cancer, but it's both of these are affecting your personality and the environment. We want to decouple the effects of your personality and the environment on both smoking and whether you get cancer, and estimate just this effect of smoking or exposure X variable on cancer, the Y variable. And we could use this causal effect to approximate the counterfactual scenario. Like if Max started smoking, you'd have a 200% higher chance of developing lung cancer, or even we might want to be able to make binary predictions like Max will or Max won't get lung cancer. And that's the most useful. And how we typically approximate these causal effect sizes is using like regression-based methods using statistics. And so I think most people are probably familiar with linear regression. We would regress some sort of Y variable outcome on X, the exposure while holding other relevant variables constant. So we have an equation like y equals beta x plus epsilon, or the error term. In this case, the beta coefficient gives us our effect size. We can make statements like a 1 unit increase in x corresponds to a 1 unit increase in y. Or if max changes, or if we go from 0 for smoking status to 1 for smoking status, we get this much higher risk in cancer. And we can also use other regression summary statistics to summarize the strength and reliability of these estimates. But you get into issues there. There's a lot of pushback because correlation and regression slopes are not causal. It's a statement that gets thrown around a lot. It's that correlation is not causation. But I think it's really important to step back and address reasons why correlation would not be causal. And a lot of this is fundamental to a lot of causal inference work because we have to use statistics in a lot of these cases. So one thing that gets brought up a lot is that random chance. What if these are just randomly associated? So to give an example of this, if we take an x variable and a y variable that have no association between the two of them, and we simulate this thousands of times, and we run association tests, and we see how often do we get significant relationships, and we look at the p-values between x and y. Based on the standard p-value cutoff, alpha right here, which is 0.05 in most life sciences fields, 5% of the time we'll see two significant associations between two unrelated variables under the null distribution. But the thing is, alpha is a controllable parameter. We set our willingness to deal with random chance in these results. So that is something that we can control. And then also another example, or another reason that correlation could not be causal would be confounding. So you'd have a third variable driving both x and y. So in this case, x does not cause y, but this z variable causes x and causes y. And one thing we can see from simulations that as we increase the strength of this confounder on both x and y, we increase, if we ran a linear regression on y as a function of x, the strength of that beta coefficient. So we would see stronger and stronger associations between x and y. And that is a major reason that correlations would not be causal. And that's something that we can identify in data and from knowledge. And other reasons that correlation would not be causal would be mediation, where your x variable is causing a mediator, which then causes your outcome. And this is causal, it's just not direct. And we have techniques for quantifying the strength of these mediations. And then also reverse causality is another reason. So if y causes x instead of x causing y. We can also use methods like time lags to see which comes first. And there are ways to address each of these. But I think it comes up a lot. If correlation is not causal, why would we use statistics or correlation at all? And something that's come up, I think a lot of people in life sciences tend to say, like, if you can see something, you know it's causal. So if we can see that like a fox might eat a rabbit, we know it's causal. But we might want to know things like, how many rabbits does a fox eat? How often does a fox eat a rabbit? What are the dynamics of foxes eating rabbits? Because there's situations where foxes might not eat rabbits, and there are also situations where it might be time dependent. It might be dependent on like prey availability and seasons and things like that. So it's important to actually use these to estimate the strength of causal effects, understand what's happening. And sometimes you run into systems that aren't manipulable, and you want to study those. So we might want to know, how does like the increasing the fox population affect the rabbit population? But it's not reasonable to introduce foxes to Rocky Mountain National Park to see what happens to the wild rabbit population, because there's a huge risk of damaging the ecosystem. So we would try to use statistical associations to try to model the dynamics of this system. And there are lots of other questions that we might want to ask that can't be experimentally validated or directly observed that we would want to use statistical models for. So one example would be, how does gene expression change in Max's lungs after he starts smoking? And we can't observe this without killing Max. And we also can't require people to smoke cigarettes to see what that does to lung cancer rates, because it's not ethical to require people to do things that might cause them harm in scientific studies. And then we can't ask massive economic questions like, does citizen quality of life improve if we increase the federal income tax rate to 90% and reallocate those funds to public goods? Because it's not feasible to run that sort of massive study. And that's where a lot of these techniques come from, is like larger scale econometrics, where you can't manipulate the like federal or global economy. And so I just wanted to kind of outline the process that we typically will use for using statistical associations for causal inference in systems that we can't intervene on or directly observe. And this typically looks like we identify variables that would causally affect systems. We might diagram them out in something like this that I'll talk about a little more. It's called a DAG, or a directed acyclic graph. And in that system, we want to identify which variables are confounding our results. So in this case, we would identify that personality and environmental factors are confounding the results, and we'd probably get a little more specific than that. And then we would address these confounders statistically or in experiments through like a randomized controlled trial or through regressions that includes these relevant covariates. And just to walk through what this process looks like, we create these directed acyclic graphs where nodes are variables of interest, edges are directional and represent causal effects. So in this case, this variable X causes Y. And also, as the name suggests, we can't have cycles because something can't cause itself. And some examples of how this might grow and what the variables in these look like is we typically have an exposure or X variable and an outcome or Y variable. So in this case, we're interested in how does screen time causally affect obesity. And here, there might be some sort of mediator as we grow our DAG, where we could add some third variable that is caused by X and causes Y. Or there could be confounders as well. So parental education could cause screen time and cause obesity. And there can also be colliders, which are variables that are caused by X and caused by Y. And once we diagram out these DAGs, we can identify patterns that can lead to spurious correlations between X and Y, or alter the correlation between X and Y, which in this case is only confounders. Colliders do not lead to spurious correlations between X and Y, and mediators also cannot necessarily lead to spurious correlations, but they do create different relationships between data. But the main variables from these DAGs that we identify are confounding pathways, and we would want to control for them experimentally or statistically. And a lot of the causal inference field has developed methods to control for these. One of the really common ones is to condition on covariates. So in this case, we would include these confounding variables, like if we go back to this example, parental education, in our multiple regression models. Another way that we could do this would be to stratify our data analysis by parental education or the confounders. We can also use more matching or weighting type methods. So if we could only consider samples, including samples that exist in similar covariate space between exposure groups. So if we're interested in the effect of higher or lower screen time, we only include samples that exist in similar like parental education space. Or we could give higher weight to samples that are similar in that covariate space, so make them affect our linear regression slope more. And then there's also a lot of really cool methods that have come out of this sort of work, like natural experiments, where we use things like regression discontinuity. So if there's some covariate gradient, like blood pressure, that affects the outcome of interest, like if you have a heart attack and you only receive some treatment once you hit 150 in your systolic blood pressure, then you can see that we would have imagined that your risk of cardiovascular event continues to increase, but this discontinuity in that slope could give us the treatment effect of receiving whatever treatment happens at 150 systolic blood pressure. And a lot of this is like this sorts of thinking about confounders is what motivates randomized controlled trials, so we can address these things through actual experiments if that's possible. And then there's also instrumental variables, which is a large part of what one of the recent Nobel Prizes in economics was awarded for, where you pick out another variable that basically doesn't have confounders and use that as a proxy through some like multi-step regression methods to try to get approximate causal effects. But in summary, We do often need statistical approaches for working with data and trying to make causal effect size estimates. There is a lot of knowledge curation that's needed to move beyond association testing to potential causal mechanisms. And the field of causal inference has a lot of techniques for this, including the use of graphs to identify relevant variables and statistical and experimental methods to approximate these causal effects and control for relevant variables. But what happens is that there's a lot of human curation going on in this. So this could be like one grad student is interested in the effect of screen time on obesity. They start thinking about what other variables affect the system and trying to grow these DAGs. So their DAG starts simple and it grows in this larger DAG. But if we start thinking about really high dimensional systems where there might be lots of variables that causally affect things, and lots of variables that we can measure that causally affect our system, it's not really reasonable to expect analysts to measure or define or manage all of these relevant variables, especially when there might be thousands. It might create this massive DAG that someone can't even draw effectively. And so it kind of leads to the question of if there's a systematic way to represent this knowledge. And I think that's where Brook's going to come in with talking about knowledge graphs some. But if anybody has questions on some of the causal inference background, we can talk about that now. Or
John,
just like one or two questions.
ever since I got my fictional cancer, it's made me want to smoke more cigarettes to deal with the stress, breaking the acyclic assumption. So why is it reasonable for you to assume that probabilities have acyclic relationships?
Because we're typically thinking of a value of a variable at a time point. So we could say like Max's smoking status last year causes cancer, which at like in December of last year, which then causes more smoking in January of this year. So typically like it would be like screen for these like to break cycles, you basically assign time points to things. So like screen time at time 1 leads to obesity at time 2, which leads to more screen time at time 3. Yeah. People have any other questions on some of this causal inference background before we jump into knowledge graphs? Cool. Yeah, so like I said, this kind of begs the question of is there some sort of systematic way to represent this knowledge in some graphical form?
Yes.
That's where Brook comes in.
And it turns out that there is a systematic way to represent these DAGs, and that is knowledge graphs. And so I'm going to give a little introduction to what knowledge graphs are for those of you that aren't familiar. A knowledge graph is essentially a collection of assertions or facts about things that are represented in the form of a graph, where nodes represent the concept and edges represent the relationship between those concepts. And here I have an example of a biomedical knowledge where you can see that the nodes are different biomedical entities, like a drug or a disease. And the useful aspect of knowledge graphs is that they represent this information in a logically consistent manner. And so because of that, and because it's this collection of DAGs, you're able to then utilize those for different questions or inferences. So knowledge graphs, again, are able to harmonize a lot of disparate data sources. And so they're able to pull together information that's represented in many different formats. So if you can see here, one of the ways that knowledge graphs are generated is from free text or published literature, for example. Another example of that is an electronic health record. So this is just prose that's in the form of a paragraph or a paper that you can access via manual curation. So you can have a group of curators looking for specific sentences that make assertions about something, about a relationship between things. Or you can utilize NLP-based methods to, for example, perform named entity recognition and identify the biomedical concept and the relationship between the two concepts. Another way that knowledge graphs are created is from structured databases. So think of things that you can access with an API or from tabular data. And ultimately, experimental results can be represented in both of these ways, in literature or in structured databases. And graphs really allow you to harmonize all those resources and standardize them into one representation of a graph. And so, next slide. Again, the benefits of knowledge graphs are threefold that I'm going to walk through. First, is that they're standardized and interoperable. And what I mean by that are first that knowledge graphs allow you to standardize terms by grounding them in ontologies, which are formalized representations of, for example, biological concepts or whatever concept that you're trying to represent. And then additionally, to the concepts grounded in an ontology, the model of that graph can also be grounded in a specific data model. And that allows you to bridge these different ontological domains and allows you to represent all of this heterogeneous information in a way that you can make use of a graph represented in another field and bring it into the graph that you're interested in. And so you build these graphs based on a particular question of interest or a particular field of interest. Second, graphs are heterogeneous, and that just means that you can represent a lot of different kinds of data with graphs by grounding them in these specific ontological terms. And then lastly, because of their logical consistency and because of representing them in this format of a data model, you're able to do inference on them. And so you're able to pull together all of this relevant information and make novel inferences about what's going on, about something that you previously didn't know about, because now you're bringing all of this different data together in the form of the graph. Next slide. And so just to give a little more of a definition of what ontologies are, these are curated hierarchical representations of terms or concepts. And so on the left is an example of the chemical entities of biological interest, or CHEBI, and on the right is an example of the gene ontology. And so these are two biomedically relevant ontologies that represent chemicals or gene and gene functions and those hierarchical relationships. And so these are curated by a team of experts that know about these kinds of biological concepts and allow you to then ground things in what everyone agrees on is the correct definition of that thing. And so you don't have these inconsistencies of what a particular biological chemical is. You just say it's grounded in this term of this CHEBI ontology. And this allows for reproducibility of research. So again, that you can confirm that the concept you're trying to represent matches what the community consensus view of that concept is. And then additionally, as I mentioned, the interoperability between graphs and between different experiments and different data structures. And so next slide. So another way that graphs allow for this interoperability concept is through their data model. And so in this slide, I'm presenting the BioLink model, which is a data model that was developed in 2022 to help standardize some of the concepts and some of the relationships between biomedical concepts. And so the BioLink model on the left, you can see that it helps define what things are. And so it provides a specific definition of what a drug is or what a drug exposure is. And then on the right, you can see the hierarchical nature of the relationships between biomedical things that have been teased out by experts and their understanding of these biological processes to say that when someone is trying to build a graph based on this model and what they know about their interacting systems, they can align to these kinds of relationships and make sure that it's explicitly mentioned in the BioLink model and it aligns with other graphs that are utilizing this model. And so what the BioLink model allows us to do is again, it's expert curated and it's open source. So it's constantly iterated on based on the use cases of people building graphs using this model. So it's up to date with the latest information about how we structure gene-gene relationships, for example. And then it's also built off of or based on the LinkML modeling language, which allows for a lot of these models to be distributed in different languages. And that just makes it a lot easier to represent different data from ontologies or from experiments and papers or from structured databases that are all in different formats to be expressed by this BioLink model. So it really helps with this interoperability concept so that graphs can represent a range of different biomedical data or whatever data that you're trying to represent. And so how do you go about constructing these knowledge graphs? This is kind of a simplified version of it and seems trivial, although each step is loaded with challenges and ways that it can become really complicated. And so the first step is essentially just to extract the data from whatever format that it's in. And so again, this may be the form of unstructured text and literature. It may be in a flat file. It may be via an API where you're querying some structured database. And then once you have those concepts and those relationships, you have to map them again to standardized identifiers. And so this step can be done with the use of NLP or the use of LLM, large language models. But generally, it often needs to be done manually to, for example, if you have a metabolite that or a chemical of interest that was named something differently in an experiment than it is in the Kebi ontology, then you kind of have to understand the context of that result to figure out which term it should be mapped to. And so this step can be really challenging, but really important to be able to access all of these different experimental results or different databases in the same context and utilize them for these purposes. And then next, once you have those terms grounded in ontological concepts, then we'll transform the assertion of interest into a semantic model. So this is, again, like saying a sentence that says this gene affects this disease. You transform that to, for example, the biolink model, and you say, this is the biolink relationship that I'm going to ground this in because of my understanding of how this is described in the text. And then lastly, merging all of those sources together and removing duplicates so that you end up with this one pretty large graph. So these can range from 5 million edges to 50 million edges in some cases, depending on just how many data sources you're trying to ingest. And at the bottom here, I've put a couple tools that help you do some of this. So the knowledge exchange format, KGX, is a Python package that helps you operate between graphs with ways to ingest ontologies or to merge different databases and removing duplicates and things like that. And then it's often helpful to build these graphs with the use of a workflow manager like SnakeMake because these steps are so critical across a lot of different sources. Okay, next slide. And so I talked a little bit about the heterogeneous nature of KGs, but to go through an example, this is KG COVID-19, which was developed in the heat of the pandemic in 2021. And what they sought out to do was to say, how can we identify drug targets that were previously unknown that may help us treat COVID-19? And so on the right, you can see a figure that's kind of the data schema of what's represented in KG COVID-19. So in the middle circle is all of the different biomedical ontologies. So you have the gene ontology, the Mondo disease ontology, Kebi, as I mentioned, of chemicals, and the human phenotype ontology. That helps you ground all of these different relevant terms. And then on the outer rim is all of the unique databases or sources of information that are critical to help us understand potential gene targets here. And so what KG COVID-19 did was allow you to address, pull together these siloed databases that were otherwise all in different formats and all updated at different stages. And then it also supports, because you've already ingested these databases, you can very easily support the frequent updating of those databases to maintain the most up-to-date information. Next slide. And so I'm going to walk through a couple ways that you can perform inference using these knowledge graphs. So that's, say, coming up with a question and then using the knowledge graph to help you answer that and get new information from it. So one is question answering, like I just said. So these are performing simple queries on the graph to say, I have this very straightforward question. What are the answers of that based on the knowledge represented in the graph? And then the second way of doing that is more machine learning-based methods where you can embed your knowledge graph, make use of the structure, and make more predictive insights as to there's a potential link between these two concepts based on the structure of the graph. And then lastly is more of a semantically constrained search. So based on my knowledge about this field, I know that things interact in this way. So you develop a set of patterns or templates and then walk through the graph and find all representations of that pattern to say, these are the causal assertions that we can find from this graph. So just to go through a couple examples of those, for question answering, this is an example over that KG COVID-19 graph. So they were able to look at, for example, what are the drugs that interact with a human protein where that protein also interacts with SARS-CoV-2. And then additionally, kind of an indirect interaction between a drug and SARS-CoV-2 is drug interacts with a human protein that interacts with another human protein that interacts with SARS-CoV-2. And so they can basically query this large graph, all the knowledge that they brought together from different data sources, and then come up with this list and validate that then with the experts at, for example, the National Virtual Biotechnology Lab collaborators to say, are these worth pursuing as potential drug targets to treat COVID-19? And then just to give a little bit of background on what graph embeddings are. So if you take the structure of the graph and you perform a series of random walks through that graph, you can come up with vectorized representations of each node in that graph that says, okay, based on these random walks, this node is really close to this node in graphical space, or really far from this node in graphical space. And you can parameterize a whole bunch of things, how many walks you take or what dimensions you want those vectors to be. But once you have that high dimensional representation of each node, you can then view this in a 2D or a 3D space and perform different vector algebra techniques to say which node is closest to COVID-19 in this knowledge graph space, which drug may most likely be interacting with COVID-19 in some interesting and relevant way. And so when you develop this set of graph embeddings, an example of using those in a biomedical context is, this is Tiffany Callahan's work from 2022, where she built the biomedical knowledge graph phenolator and was able to represent patients in the form of vectors by vectorizing her graph. And so when you first take clinical embeddings, where these are kind of embeddings based on the free text from an electronic health record, and then you take the embeddings of the phenolator knowledge graph space, you can say for this patient, based on these different phenotypes, for example, asthma or cough, you can take that patient's specific clinical notes and represent them as a vector as how those specific phenotypes are represented in the graph. And then you can make inferences as to say this patient is most likely had this rare disease, for example. And this is what Tiffany Callahan related this to, was to say rare disease is really hard to figure out. And so because it's rare and you don't have a lot of information about it and you don't have a lot of insight as to which symptoms are associated with which rare disease. And so when she applied this to, for example, cystic fibrosis or sickle cell disease, she was able to essentially computationally phenotype these patients and say, based on these electronic health records and based on what we know about this biomedical data, we can then say this patient most likely has this rare disease. Next slide. Okay, so just kind of to sum up background of knowledge graphs, they're semantic representations of knowledge. And that means that you can harmonize a large amount of data, and you can also bring together a lot of varied structures of data. And that's critical to John's question of how can you move beyond just manually curated simple DAGs and really use more of the biomedical knowledge that we have. And then additionally, because of the way that they're represented in the form of a graph, they can be queried. And that's really great because you can ask questions and get out novel inferences about the questions that you have. And so we kind of showed this in understanding SARS-CoV-2 through the KG COVID-19 graph that I talked through, and kind of getting at novel drug discovery to address that disease. And so now we're going to talk a little bit more about what are some other biomedical applications of knowledge graphs, and how can these be shown to sort of get at these causal mechanisms at play. So yeah, any questions on knowledge graphs? Sweet.
Cool. Yeah, and I like Max's responses in the comments about cycles, but we can talk more about that at the end if anybody wants to. Yeah, and just to get a little bit more into the biomedical applications, Brook and I both study the gut microbiome along with Kathy Losbone, who is our advisor, who is here. If anybody wants to ask Kathy any questions as well at the end, surprise Kathy. But just to give some introduction, we all have digestive tracts where we eat food and digest and absorb compounds from that. And in our digestive tracts, typically towards the end, we have a community of microbes that lives there. And these help us break down food. They also, sorry. They also have other relevant functions, like they train our immune systems as we develop how to deal with microbes. And they also have other functions, like they help prevent infection from different pathogens. And just to give an idea of the scale of the healthy human gut, typically has roughly as many microbial cells as we have human cells in our entire bodies. There are over 1,000 species of microbes present, and there are over 150 times more unique microbial genes than human genes in our guts, which is pretty important because genes in the microbiome are relevant to human health, so they affect whether or not we get diseases, how our bodies respond to things, what we get out of our foods. But unlike the human genome, we can actually modify the microbial genomes that we have and what they're capable of doing. The human genome cannot actually really be modified while you're alive. How the genes get used can, but it is really interesting that these genes are plastic and modifiable, and eventually we can have more interventions related to them. And we give an overview of how we study the gut microbiome. We typically use really high-dimensional genetic and chemical probes. So you can imagine your diet, environment, and other factors like genetics impact the microbes that are in your gut. These microbes have DNA that make up their genomes. We can sequence these, and that's called metagenomics. And kind of the central dogma of molecular biology is that DNA gets copied into RNA, which is kind of like a single-stranded copy, which then these RNA molecules make proteins that have some biological function that create or alter metabolites or compounds or molecules. You can kind of think of them as just like these are things that do things in your body, and that impacts human health. And we can kind of measure it at each step along here. And when we measure these things, we get roughly 1,000 to 100,000 features. 100,000 would be a lot, but somewhere in that mix, you might see really commonly like 40,000 feature data sets. We often have multiple data sets per study. So if we want to study some disease, we might do metagenomics and metabolomics. So this step and this step, and that would be a total of over 50,000 features, maybe nearing 80,000. And it's really common to try to do like biomarker discovery type research. So what's really predictive of a disease or some sort of outcome, what predicts drug response, what could be used as a potential therapeutic. And in this case, I'm just going to walk through like an example data set and study. This is what a lot of my dissertation is focused on dealing with, which is taking a piece of this gut where you can imagine the human digestive tract is here, and you have a community of microbes that is sitting right by it. And they're kind of sending messages back and forth. The microbes might eat some food compound and produce some other food compound that then the human absorbs. And they're in constant conversation at this surface. And we take a sample that has both the microbes and the human, and we sequence what genes they're turning on at any given time. And from this approach, we might have 40,000 human genes and 40,000 microbial genes in a data set. An example question that we might want to ask with this is, for each gene, what other genes causally affect its expression? So to put this in other terms, how are microbes and human cells communicating with each other molecularly at this surface? Or how are microbes regulating what they and other microbes do? And in this case, we have 40,000 microbial genes and 40,000 human genes, so it's 80,000 total genes. And we want to model every pairwise combination of genes and get at what gene causally affects what gene. But in this case, that has 3.2 billion pairwise combinations of genes. And typically that can be problematic for analysis reasons because it's a large number of combinations to be looking at. So in a lot of these cases, we want to control for the number of false positive associations that we find, which means that p-value threshold for like random chances of associations that we're interested in gets really reduced, which doesn't give us a lot of power to detect statistical associations. Makes it really hard. And typically in these cases, we would want to identify confounders. We want to run statistical model diagnostics. And a lot of these statistical models are not using standard regressions that are fast to run. They might be using like Monte Carlo Markov chain-based sampling, which is a lot more expensive and slower. And when you're doing that for 3.2 billion regressions, it ends up being a lot if each one takes like a couple seconds each to do all of these steps. And just on top of the time and statistical power and whatnot, it's just impossible for a human to catalog the confounders in 3.2 billion relationships between genes. We're going to know which ones we should plausibly be testing, which is where it kind of comes in of if we can use knowledge graphs to help with analysis of high-dimensional biomedical data sets. And there are a few questions that we kind of have to be able to answer with that. like, do we have knowledge graphs that synthesize what we know about human and microbe interactions, or microbe or human cell to human cell interactions? Can these effectively be queried for patterns of interest? And then there are other questions like, can we use these knowledge graphs to effectively guide us statistically? So we have these knowledge graphs, but can we reliably identify which genes are relevant to study, or which genes are confounders of like the relationship between, say, gene I and gene J? And Brook has done a lot of work on talking about this first point. Do we have knowledge graphs that synthesize knowledge about human microbe interactions that we can query for patterns of interest? So she
Yeah,
can hop in here.
so my dissertation was really focused on this question of A, do we have these knowledge graphs and can we use them to address mechanistic explanation, microbial interactions in disease? And there weren't there was a lot of potential to develop these knowledge graphs in the microbiome space. And so I'll walk through one of the examples that I did in my dissertation for this, which was developing a knowledge graph called microbe gene metabolite link, or MGM link, where I integrated the gut mgene database, which is a database of correlative relationships between microbes and human genes, as in microbes that regulate gene expression, and microbes and human metabolites, or chemicals of interest in human metabolism, and how they influence the amounts of that metabolite in their system. And I represented this in the way that I could combine it with the biomedical knowledge-based phenylator, which brings together a lot of human focused information. And so can we move from just not just microbe to the genes that they interact with, but microbes all the way through to the ways that they may influence different diseases? And so using this knowledge graph, I developed two different search methodologies. One that was based on the structure called an all shortest path search, which was just saying what is the shortest distance between node 1, which would be the microbe of interest, and node 2, which would be the disease of interest. And then one that was more of a template-based search, which I mentioned prior but didn't get so much into, which was, can I develop these templates and identify all of the ones that exist in the graph that have this microbe of interest as the source node and the disease of interest as the target node? And from all of those paths, you come up with a lot. It's a really big graph. It was a graph of 5 million edges or so. And so I also developed a path prioritization technique that, based on those graph embeddings, which paths have nodes that are most similar to the disease of interest. And it turns out with these paths on the right that I was able to find feasible mechanistic explanations of the ways that, for example, F. prausnitzii influences Parkinson's disease through its interaction with immune markers, or the way Streptococcus influences inflammatory bowel disease, again, through the ways that it interacts with the immune system and other metabolites in the human body. And so this methodology allows us to start to get at this question of John's, that we found that we could kind of combine these two methodologies and say, like, here's how we can start to find these causal interactions. And so now what I'm pushing more towards is developing this graph that represents functional annotations of the microbes. And so for John's question of these microbial genes and these human genes, now we have a graph that represents both microbial microbes and the genes that they express, humans and the genes that they express, and all of the different functions that those genes have, and then utilize that semantic structure to start to understand the ways that those genes interact and the ways that they influence disease. Next slide. When we ask the question of how knowledge graphs may be able to guide us statistically, as in which genes are most relevant to the study, or what are the confounders of this gene versus this gene. And we can look at this study that was performed in 2023, where what they did is they developed a way of kind of finding these causal features in a knowledge graph. So they scoped out the biomedical literature and found different assertions of a disease-specific question. They looked at Alzheimer's and depression. And using those, they mapped them to a biomedical knowledge graph phenylator, which I talked a little bit about before. And then using the structure of the graph and the semantic patterns, or DAGs, they found different potential confounders, mediators, and colliders in that graph that you could then go statistically validate using the methodology that John was talking about. So again, in the next slide, they applied this methodology to depression and Alzheimer's disease, mostly because, first of all, these two diseases are multifactorial, or they have different genetic and environmental risk factors. They're heterogeneous, so they have varying array of symptoms and severity. And then furthermore, they have overlapping risk factors. So that means that this group expected that, well, we might find concepts that play multiple causal roles. So can you use the graph to help you find what role those factors play? And additionally, with depression and Alzheimer's, it's really hard to study these diseases because again, there are a lot of modifiable risk factors attributed to dementia or Alzheimer's disease, and that includes depression. And so if we can figure out the ways that depression may be confounding in this disease, can we reduce the prevalence of depression and therefore reduce the prevalence of Alzheimer's disease? So this was a really nice use case to ask these kinds of questions. And so what they were able to find was, this is kind of just the list of all of the different confounders, mediators, colliders that they found. But ultimately, they found 11 variables that fulfilled criteria of all 3 causal roles. And that basically means that if you look at all the different DAGs represented in phenolater by the information that they mapped to that graph, then you can go and start to say, like, okay, what are the statistical tests that I can run to really figure out what the interactions of these concepts in this space really are? So ultimately, this methodology helps them pull the critical information from a large amount of biomedical knowledge to say these are the ones that we should really test.
And just to hop in here, I think this does kind of get at also some of the limitations of using knowledge graphs in these cases. And like Max put in the comments, you need to be really careful about connection types to avoid like bidirectionality or cycles. Like in this case, I mean, theoretically, something cannot be both a collider and a confounder in like a true ideal causal inference DAG scenario. But in the way that we represent knowledge, like if you're systematically searching this, we haven't quite identified yet the right criteria for this, or it at least requires some sort of human intervention at the end to curate it. Yeah, and so just to like map out what an example workflow looks like here, there's just kind of 3 main steps: is to take our data set and ground it in the entities represented in our graph. So we might have this list of 40,000 microbial genes. We want to map those, ground those to some protein or some enzyme commission number here or some molecular function in our graph. We would apply some sort of template-based searching, like Brook mentioned before, where we could go from like a microbial protein that causally interacts with some metabolite or compound that causally interacts with some human protein and start looking for these DAG patterns. And then we'd use these to constrain our statistical models by plausible DAGs from the knowledge graph. And we would ideally get some sort of table of which microbial proteins and which human proteins are interesting to look at. Just to kind of summarize the benefits here, these causal inference techniques are really critical for approximating interventional effects. And a lot of these rely on graph structures to identify confounders, mediators, colliders, things that need to be controlled for. And we do have ways to systematically represent causal factors and systems, like through knowledge graphs, which can harmonize data, and we can perform DAG-like pattern queries on these. And then we can also use the knowledge graphs in parallel with statistical techniques for inference of causal effects, which seems like a really promising field. And just to go through some acknowledgements, the Lisbon Lab has been super helpful here. Brook is also co-advised by Larry Hunter. I'm co-advised by Chris Lowry, and the Hunter Lab does a lot more with the natural language processing and knowledge graph type work. And yeah, we have our funding sources acknowledgement here, and here's the Lisbon Lab. And Brook, I don't know if you want to say anything about the Lawrence Berkeley National Lab.
Oh yeah, that's just that they've been really important in some of the microbial knowledge graph development that I've worked on. So just an acknowledgement of that.
Yeah, and we can take any questions, or we have some stuff on like what an example project with this looks like. Kind of what it feels like Brook and I have done is, since we're both nearing the end of our dissertation, kind of putting these together. Here's a grant application or a PhD project. Whoever wants to do this can follow up and do this. We've shown some examples of where it has worked in the past, but combining this directly with the microbiome data sets has not exactly been done. But yeah, if anybody wants to talk more about any sort of causal inference work or knowledge graphs, feel free to reach out to Brook or me. We're both, defending this year. Open to more directions if people have thoughts or questions or just want to chat.
John and Brooke, that was super cool. Thank you so much. I have some questions, but I've already asked a couple, so I'll wait and see if other people want to jump in first.
I have a quick question. Do like moderators not tend to make it into DAGs in your field, or is that just something you excluded for no reason?
Yeah, they do, like mediators, and but a lot of times, uh, the question is, do you actually care about indirect versus direct effects through mediators? And in that case, like it's a great time to do mediation analysis. But also doing mediation analysis opens things up to more confounders in each step. So you might have something that is a confounder for your X variable on Y, but is not necessarily a confounder for the mediator on Y. Uh, and that could be, it just requires some other consideration. But it doesn't, mediators don't necessarily create false associations between X and Y, whereas confounders do, if that makes any sense. Which is why you don't have to control for mediators. It really depends on your question.
Do you incorporate like moderators as a separate thing from mediators, or are they sort of lumped together? As in like something that might, like a moderator moderates the relationship between one thing and another, as opposed to a mediator, which like is a conduit for the relationship.
Yeah. Sorry, I'm going through a bunch of slides back to, I have to go back further than I thought. Like here, so like the mediator would be physical activity, but an effect modifier would be like a moderator would be something that modifies the effect of screen time on physical activity. Oh, okay. Yeah. You that would be included depending on like if it also affects Y. Yeah. And there's also other variables like it's some variables will affect precision but not bias. So a variable that only affects your outcomes, so something that only affects obesity, will affect the precision of your estimates. And thinking more like that'll affect your p-values, but it will not necessarily bias the effect of screen time on obesity if it doesn't also affect screen time. So it kind of depends on if the effect modifier only affects screen time on, or like in this case, the effect of screen time on obesity, and what exactly you're trying to model. If you're interested in that effect modification. Yeah.
Okay, thanks.
Yeah, that was a good question. Thanks, Kieran.
So in computer science, a lot of people are keen on this idea that somehow we're going to get language models that are big and bold and brave enough that they can give us new novel ideas, right? Like novel experiments to run or something like this. I'm quite pessimistic on that notion, but I may be proven wrong. But it does seem like something like maybe some level of causal inference with knowledge graphs could allow for something like that, where you maybe are doing hypothesis generation by generating a knowledge graph and then having a language model describe in plain English what sort of hypotheses could be tested based off the shape of that graph. Have you thought about something like this before? Or do you think there's any juice to squeeze there, or is this kind of like, silly, I don't know, San Francisco sci-fi thinking?
Um, it is it is paramount to what we look at in the Hunter Lab. Actually, is like the whole question of like, can you write the discussion section and like the future direction section? And actually, a recent graduate of Larry Hunter's lab, Mela Boguslav, created an inference space where, an ignorance space where it was like everything that we don't know about things, and then let's help this guide our field into based on all these papers, what statements are made about what we don't know, and then can we kind of like figure out what we're ignorant of. And so no, you're not out of field. It's that combination though of what I have found in my dissertation work is that LLMs, of course, we all know, with hallucinations and with kind of like the way that they are trained, that you kind of need, especially for questions in biomedicine, that you kind of need the semantic structure of the graph that really says, like, we know exactly where this information came from. We have the provenance, we have the structure, we have everything we need to know that this is logical. And then, so like the KG-RAG approach, where you start to bring those things together, are probably more promising than just saying, like, let's just query LLMs for this. But it is like the future, I think, is like important to start to integrate both of those techniques. Yeah.
Cool. Thank you.
I also, just to add to that a little bit, I see that with with like drug discovery work, where people are doing a lot of like AI guided drug discovery. When these things work in like preclinical trials, and then you move on to clinical trials, and you spend a couple $1 million to run a clinical trial, and it doesn't go through, having some sort of background for the reasoning required to generate this drug that failed is really helpful to understand why it failed and how you could find a very similar target drug that could avoid failing in the next case. That I think like knowledge graphs are really useful, or having more of like the reasoning steps through a lot of the LLMs is also really helpful.
Real quick, you had mentioned that there was someone who was working on the ignorant space, I think is what you mentioned. Was there like a paper that they had released, or how can I read more about that?
Yeah, for sure. So I can also, if we're running out of time, I can also email it, Max, to you, and you can send it out. But I think this is kind of Mela's main recent paper that I'll link in here. And then this work is being continued on by Nora, who's the second author on this in Larry's lab now, to kind of start to use it for asking specific biomedical questions. So it's pretty cool stuff. Yeah. I think that's the main one to share.
Cool. Thank you.
So maybe just to finish up, if people have more questions, should they email, you two, or what's the best way to get in touch?
Email's great.
Yeah, email's great.
Okay. So I'll share your emails in the Slack, and then if somebody watches this and is not a member of the Slack and wants your emails, they can reach out and I'll add them.
They're also, I think, on the slides if they watch it and want to go back to the last slide.
Awesome. Well, Brook and John, thank you so much for joining us today. Super fun. And working on something really interesting that kind of scratches the itch of like actual thinking that the LLMs don't do. So as somebody, dealing with the other side of things all day, I'm very excited to be hearing about more semantical reasoning. So I appreciate you taking the time on this Friday to chat with us.
Yeah. Thanks for the opportunity. Thanks for joining, everybody.
Yeah, thanks, Max.